This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 68, recorded on November 7th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hey, how are you?
Welcome.
All is well?
Here as well, out there.
Fine.
I don't know, what are you, 3,000 miles away? It's pouring here, though.
Oh.
And it was really noisy, but it stopped. See, the storm passes up the... I have a view of the Hudson River here.
No kidding.
From my office, the George Washington Bridge, and I can see the storms moving up the river. It's really impressive. I don't look forward to retiring. I won't have this view.
Well, you just have to get an apartment in Manhattan on a penthouse, so you can.
Oh, yeah. On a retired professor's salary. That's happening. I'll have to write a textbook. How's that?
I don't think that's going to help.
Also joining us today from Medical University of South Carolina, Michael Schmidt.
Hello, everyone.
How are you doing down there? Is it raining down there?
No, it wants to, but I'm just back from a site visit at Washington State University where I can report they had 3 inches of snow on Tuesday.
Yeah, it's winter in some parts of the world, yep.
So I got to reacquaint and activate my Chicago genes of driving in snow. And, those things that you learn when you're young actually come back pretty quickly when you're old.
That's right, when you need them. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello, everybody.
How are you doing?
Great. I can report that it's spring in other parts of the world.
Spring in Michigan?
No, but in Australia. I returned from the Legionella conference that we have every 4 or 5 years. Really? And it's beautiful there.
That's right. The last time we did this,
Where
you were heading
in
out
Australia?
there. Melbourne.
Oh. I'm going there.
That's a beautiful city.
I'm going there next July.
Oh, good.
Which is winter, unfortunately.
Winter.
Yeah.
Mm-hmm. You'll have to go see the penguins.
Yeah?
It's a treat. You can go out to Phillip Island, and at dusk, once it's safe, the penguins come in from the ocean from having fished, and they, once they get a little quorum and courage, they trot across the beach. It's fascinating to watch.
A quorum. That's great. Did you take pictures of it?
You're not allowed to take pictures because the flash damages their sensitive eyes. But you can go on the World Wide Web and find it.
Wow.
Yep.
Yeah, I'm going to the ASM meeting next year, which is the Australian Society for Microbiology, right?
Yeah.
Which is in Melbourne.
They were advertising that meeting at our conference. So.
Is that your first time there?
It is.
Yeah, I haven't been either, so.
Hopefully not my last.
So the flight is horrible, right?
You just think about all those miles you're racking up.
Yeah, right. Hey, did you play golf there?
I did not. I was too busy enjoying the local flora and fauna.
Okay. I would, if you really were a golf nut, you would have brought your clubs, right?
Nut. Yeah, emphasis on nut in that case.
You're not a nut. All right. I have a couple of papers for you today, but before we do that, Elio, how is your poll doing over there on the microbiome?
Well, you had the idea of polling the folks, so I did that on Small Things Considered, and we got quite a few responses, and about 2/3 like the idea that the term microbiome refers to the bacteria. 1/3 roughly like the fact that it refers to the genes of the bacteria. And I got some nice comments, and I'm analyzing them, and what I can tell you is that, unfortunately, this did not solve the problem. I didn't think it would. In fact,
Vincent
I think we're back
would.
on square one. The opinions are divided. And people bring up some interesting, in fact, amusing facts, like the fact that the word microbiome can be parsed in 2 ways. Micro and biome, and that's clear enough. In a biome, I think A biome is the whole thing. A biome is the aggregate of the environment, and it's biota. It's sort of what I think that Michael will like that. That's what you said.
That was my view.
Okay. The other way of parsing is microbiota. Now, ota then becomes like, everything else. Microbioma.
Oh.
Becomes an ohm. So now an ohm becomes like a genome or proteome and so forth, and that sounds more like it's the gene. So that's a curious semantic way of looking at it. I thought it was useful. It was amusing.
Cool. I hadn't thought of that, yeah.
Some other folks had some interesting things to say. So you're gonna see this. I'll just I'll publish this very soon. It'll be posted in a couple of weeks.
There's some interesting comments.
I don't know where to go with this. I mean, do we want the ASM to be the arbiter on this? I mean, the use of the two terms is distinct and bothersome to me that the same word means two different things. So I have no idea who can...
I elect you, Elio. You be the arbiter.
It could be the arbiter.
Well, it brings up a really important point because, as we're seeing the literature evolve and as I'm being asked to review internal grants here at the university, a lot of folks are beginning, who aren't practicing microbiologists, are beginning to employ the techniques that are commonly associated with analyzing the microbes associated with animals. I think that's the safest way to operationally define it. And not only are we having issues with proper use of the word so that you know what sort of science they are conducting, but we're also similarly having issues with the best practices for the techniques to address a particular scientific question. So this is one of the functions of the American Academy for Microbiology, where they begin to come up with best practices of how to practice the art associated with the science. And this could be, the thesis statement of a colloquia: what is the microbiome and what are the best practices or techniques used to investigate questions of the microbial interactions with the host.
You should suggest that.
That's a great idea. That's a great idea. I think that's very appropriate. Okay, I'll be glad to pass the word.
Pass it on. And then
Bonnie Bassler is the chair of that fine group, so of the Board of Governors.
Right. Bonnie can be the arbiter.
Or organize the discussion.
I'm kidding.
You'll get mail.
I like this comment and the comments on your poll over at Small Things Considered. I'm still working on gene and epigenetics.
Yes.
I think that's great.
That's a great comment.
We haven't settled a lot of things, right? Okay, we have a couple of papers for you today. Elio, would you like to lead off?
Sure. Okay. The paper that I'm going to discuss is called High Level of Intergenera Gene Exchange Shapes the Evolution of Haloarchaea in an Isolated Arctic Lake. And it's got about a dozen plus authors, mainly from Australia. And not the same folks that dealt with Legionella, I don't think.
How do you like that? How do you like that PI's name, Elio? Cavicchioli.
Cavicchioli, very nice name.
Is he a relative of yours, Vincent?
We all are, aren't we?
Yeah.
Anyhow, they are mainly from Australia, but a few are from Los Alamos and from the Joint Genome Institute in Walnut Creek, California. Anyhow, this is a very interesting study because it has a great deal to say for itself from a touristic point of view. That is, if you want to find a lake where the temperature is well below freezing, something like minus 20 degrees below freezing, and it's still liquid. And you're going to find this in a very few places in the world, and one of them is in Antarctica. It's called Deep Lake. The reason it's called Deep Lake is because it's deeper than a number of other lakes which are also around there. And the reason, of course, they are liquid, despite the low temperature, is that they're hypersaline. 10 times the salinity of seawater, which amounts to 270 grams per liter.
Wow.
Salt. Okay,
That's a lot of salt.
Wow.
That is salt. So if you could swim there, you would be floating. That's the good part. I don't know that anybody has done it because you'd have
Your
to
dead
have
body
quite...
would float.
Oh my God.
Yeah.
Right. Anyhow, this is part of East Antarctica, an area called the Westfold Hills. And there's a bunch of lakes there, a few of which are hypersaline and probably still liquid. Now, what's amazing about this place is that it has archaea in it, and a few bacteria and some algae, mainly one kind, the Dunaliella, which is apparently the primary producer of organic material. Very little of it. This is as close as it gets to the place on Earth that has liquid water and has few bacteria. Mm-hmm. It is said to be the lowest productive lake in the world, known among at least liquid water lakes. So if you ask the question, I once asked this question in our blog as a Talmudic question, are there any places on Earth which have liquid water and which do not have microbes? And I think nobody has come up with any so far. And this is as close as it gets. That is, it has microbes in it, but very, very, very few. I have a hard time finding the actual amounts, but it is less than 10 to the 5th cells per liter. That is less than 1 per ml. That's on the surface. On the bottom of the lake, it may be about 1 cell per ml. That's not a lot. But they're there. They're there. And they do something rather remarkable. Let me tell you the punchline, and then we'll go over it. These guys have one of the greatest rates of exchange of genes known anywhere. So they exchange lots of genes, and they exchange big pieces, up to 35 kilobase in length, and essentially 100% conserved. That is, you find exactly the same piece, 35 kilobases long, in one species as in another. And what you find is essentially 4 species. 4 dominant haloarchaea. Haloarchaea are the salt-loving archaea, like Microbiome and many others. They're well known from the temperate and tropical zones. They're somewhat known from the Arctic and from cold-loving sites, but it's very little. Anyhow, to show you that life can be persistent, not only do you find bacteria, but on the shores, you find some skeletons of, or if not skeletons, mummified remnants of penguins, seals, tube worms, and a few things like that, and shells. So around there has been life at one point. Right now, there is very, very little. So these guys, these halobacteria, they amount to 72% of the whole biota is halomicrobiome. So it's not exactly a pure culture, but it's pretty clean. Anyhow, they grow. All these things, the 4 dominant haloarchaea they have been given some. They're not known, so they have been given letters or other names. One has a name, Halobiome locus profundis, which means deep lake. So it comes from deep lake.
It's great.
Not bad.
They've been working on their Latin translations.
That's right. That's right. Anyhow, what's interesting about these guys is that they grow mighty slowly. They can be grown in the laboratory, where oddly, they like it warm. They grow between 1 degree and some 35 degrees, 38 degrees centigrade. They prefer, or 42 degrees, but they like it best at 33 degrees. So given a choice, they'd rather be in Florida than in the Antarctic.
It's funny. It's funny that they can grow at high temperatures, isn't it?
You know, there are thermophiles that can be found under the permafrost.
Okay.
So when talking about the convergence between physiology and ecology, it ain't perfect.
Apparently. So they say that they divide 6 times a year in the cold. 6
times a year in the cold. Yeah, they divide once in 12 hours, I think, at maximum temperature.
That is stunning, that number.
Yeah.
But I hear there are bacteria that divide once every thousands of years even.
Well, be careful. That's the average. What if they divided once every week, but they die almost once every week? So the net growth isn't much, but the growth rate is not what you think it is.
It's always about the net.
Yep.
That's right. That's the net. So if you were to study this carefully, you may find that the growth rate is considerably higher. And that may be true for the places where the generation time is said to be thousands of years. It doesn't mean that. It just means that the net increase is very slow.
How would they know that these bacteria, these archaea, divide 6 times per year? How would they know that?
That's from the laboratory. They did that's I believe it's from the laboratory.
So at very cold temperatures, they can extrapolate.
They put them at low temperatures, and they observe the growth, and the growth is very slow. And so after a month, there is hardly any growth. After 2 months, they may have doubled once.
Right.
So if you have enough patience, this is the great new thing in microbiology is patience.
But that's counterintuitive to the way we were all selected. My old advisor told us we were all selected because we were hyperthyroid, and we like experiments to go fast. Otherwise, we would be out there becoming sequoia ecologists.
That's right, exactly. But
that's good.
And it turns out, and this whole business about so many bacteria not being cultivable has to do with patience. In this case, if bacteria grow, even at best, they double every 10 hours. You don't get a colony the next day. Yeah. So, you may have, in some cases, you may have to wait months and months. And people who do that, who are looking for bacteria in the environment that are supposed to be non-cultivable, if they save their petri dishes and wrap them in some material so that the agar doesn't dry out, eventually they find little colonies. So, anyhow, that's the story on these bugs. And let me say a few more things. The metagenome from this area, that is the whole DNA from this area, from this lake, is unique. This is really a strange place. It doesn't resemble very much of anything else. By the way, these bacteria make biofilms. So that complicates your measurements, and because in a biofilm, again, some may grow faster and some may die. So it's very difficult, in fact, to determine what the growth rate really is in slow-growing biofilm formers. Now, in other haloarchaea, so the point of this paper is that these guys would go for a strange form of sex rather than for eating. They don't have much to eat, and they can't do much at the temperature and at that salinity, so they acquire genes and they exchange genes. Now, other halobacteria, the temperate ones, are known for having all kinds of goodies like viruses and conjugated plasmids and transposons and all kinds of, they recombine like crazy. So it's not unusual that these guys do too, and I believe that genomes show that they have the wherewithal for this kind of genetic exchange on a big scale. One interesting thing is, if you look, since they have all this genomic information, I don't think they've reconstructed the entire genome of anybody yet, but they have all kinds of genes. And so they find that there are probably different, there's probably an interesting speciation namely some may have gas vacuoles, bacterial rhodopsin, so they don't need to eat. They can do photosynthesis. Obviously, they have lots of ATP binding cassette reporters. They're saccharolytic, that is, they eat sugars, and the way they put it together is that the authors think that there is not just 4 different major species, but they occupy slightly different niches. There may be overlap in them, but some of them are probably found deeper. By the way, the name Deep Lake comes from the fact that it's fairly deep, as such lakes go. It's 36 meters, I believe, is at the depth. Anyhow,
could I jump in and just expand on one of those points that you made? Elio, you mentioned that they make gas vesicles, and they also have that protein bacteria rhodopsin. So I thought it was especially beautiful that they speculated that the dominance of that particular microbe was because these gas vesicles would allow it to float up to the top during the warm weather, and then they would use this rhodopsin to harvest energy from the sunlight, which of course would only reach the very top levels, and allow them to multiply at a higher rate. So, and they could migrate faster using these gas vesicles than using flagella, which would cost energy in that. So I just thought that was cool. Really beautiful.
Yeah.
It's a good point. That's right. Well, anyhow, from this, they derive a lot of conclusions about the evolution of these things and how what all happened. So I must tell you that this is I found the paper quite difficult because it goes into a lot of evolutionary theory, a lot. You have to really know, and they are not sparing when it comes to the use of specialized terms and abbreviations. I had to look up and really go look for it and find that FR stands for fragment recruitment. Well, anybody who's in the business of doing metagenomics knows that, but I didn't. I'm not in that business. So there's a lot about analysis having to do with IS sequences, with a lot of dominance. Niche adaptation is a big deal. The paper is not an easy paper, but I'm sure that it's an extraordinarily well-done paper, and that people who know more about this than I do will get a lot out of it because of the uniqueness of these organisms and their environment. So certainly
and
one of
some of their beautiful illustrations, I think, help the rest of us who are just outside the field. So figure 2, I thought, was just a really nice depiction of the frequency of one of the members of this small community to donate its DNA to the others. Yeah. So it's one of the challenges that microbiologists have now that we have loads and loads of data, is how do we illustrate it simply, elegantly?
What do you mean without the linear algebra that they presented?
Right.
Right.
The fact is that metagenomics, or the ability to find out what's there in a natural sample, has appropriately been extended to very strange places. I think Norman Pace has pioneered that with looking at the dry deserts of Antarctica again. So we're going to see a lot of this. People have the tools are there, both the sequencing tools, which allow rapid and cheap sequencing on a huge scale, and the bioinformatics, that is the ability to analyze this. So what do you think? Are we going to see much more of this sort of stuff?
Sure, absolutely. It's not so hard to do. I mean, going there and sampling is hard, I believe, but the actual sampling isn't difficult. The sequencing isn't very hard. I think the hardest part here is the analysis, but then you do that on a computer in the lab. So I think this would be done as much as you could.
Well, except it has to be done by people who understand this.
But it's really the evolutionary theory, too.
Yeah, understanding is I had a lot of trouble with this paper as well.
You did too?
It's a lot of jargon.
I'm not alone.
It's a lot of there are a lot of abbreviations that I'm not familiar with. I mean, I get the conclusions, but we really put a lot of faith in the bioinformaticians because we're accepting what they tell us that they're doing, and I'm not so comfortable with that.
Well, that's why I suggested at the first part of this discussion, having read both of these papers that we're going to discuss today, that we really need to come up with a best practices to really understand. We're the microbiologists, and none of us are, or the majority of us have not been trained in this very specialized field of bioinformatics dealing with big data. I think that's the easiest way to describe what's coming out in our discipline is we're getting a lot of data, and we really have to make sense of what's actually going on. And when we get to the paper that I'm going to discuss, I'll introduce a concept where it may apply to this one as well. And I was scratching my head when I was flying back on the plane yesterday trying to understand if the same thing I saw in the paper I'm going to present actually applied here, and that's about biomass. Species richness declined as biomass increased. And in these very extreme environments like we have in Antarctica where it's very cold and the biomass isn't very, for lack of a better word, high, you wonder whether or not you're going to see similar systems where you have a lot of diversity, but whether or not this diversity happens to be a function of this horizontal gene transfer, and does that then create a new species or these OTUs, which is an operational taxonomic unit, which is our way of saying we have identity. And I think on the last TWiM, we came up with, we have the lumpers and the splitters. Do you make a new species or do you lump it into the same and call it a strain? And, it's a really exciting time because the tools have become affordable, but now we have to begin to make sense and be able to tell the story at the end.
We should note that they collected over 20 gigabases of data.
Wow.
Yeah.
In this, they sampled from different depths of the lake and sequenced
Get
it.
used to them. Get used to them.
Yeah, it's a lot of data.
There are a few points that I think even classical microbiologists, though, can appreciate. One that I really liked is they pointed out that this high rate of gene exchange occurred in an environment that is very similar to what many students will be familiar with, which is high salt and ice.
That's right.
When we want to transform bacteria, we put the tubes on ice and we give them high salt, and that is thought to stabilize the cell-cell interactions that are needed for the exchange of DNA. So I thought that was cool. And then they also point out that many of these large regions, 30 or 40 kb, that had been transferred from one member of the community to another, but were identical, are flanked by enzymes that can mediate that excision and transfer. So these are selfish DNA elements that can promote their own spread.
Interesting.
Well, I'm glad you made that point. It's absolutely essential to understand what's going on. Thank you.
So the thought is that maybe the salt and cold is part of the process, but we don't really know, right?
Well, you're never going to know, but it's a plausible, it's a possibility argument.
Sure.
It's
plausible that these are
It is pleasing. So the idea is that because the bacteria are held together very well by salt and cold, they're exchanging a lot of genes, right?
It does favor that.
Yeah, but it's because
conjugation.
I was wondering if it had something to do with the low nutrients or the low biomass or the whatever, but that makes a lot of sense. It's just kind of accidental. So maybe it doesn't benefit the Archaea to have such gene exchanging, right? It just happens because of the conditions.
Yeah.
They make a statement here Maybe someone can explain. They say the genomic characteristics of the 4 haloarchaea are consistent with a lake ecosystem that sustains a high level of intergenera gene exchange. I got that so far. While selecting for ecotypes that maintain sympatric speciation. I don't know what
that means. You can have sex with each other.
Is speciation in one site, as opposed to allopatric, which is when, let's say, two organisms go into different places, different environments, and they speciate in different environments. Sympatric means it happens in the same environment. That's all it means.
So this is just a summary of their observations, essentially, right?
Yeah.
I think it's amazing that these are different genera, right?
Yeah.
And so
that's right. It's in the title. High level of intergenera gene exchange.
That's unusual. And so I take it from the title that gene exchange between genera is unusual, right?
Well, we Shigella and E. coli, that's how we got 0157.
That's right.
So, it's, I think, the mammalian speciation, where it's operationally defined as when you have sex, you will not make offspring. Maybe that's too far of an extreme. And again, we're basing it on our frame of reference, but it's really a microbial perspective that the speciation is actually dealing with, and whether or not the genes are compatible, whether or not the methylation will allow the nucleic acid to integrate before the restriction systems trash it before it becomes stably maintained. And then, of course, the fitness question is asked: does the new information confer selective advantage to the organism so that it will carry it forward to the next generation? Right. And that's really the key here, because of the profoundly short generation times. They may just take the selection for horizontal gene transfers, because they divide so slowly. They're not able to make the decision based on arithmetic, the population expanding quickly, which will then select out. They have to take all comers in and then decide in time which gene actually facilitates the pattern.
You're right, but the machinery that does the translocation can be quite promiscuous between genera we see here, but also, for example, similar machineries allow some bacteria to transfer their proteins directly into eukaryotic cells.
Right.
Yeah.
So the machinery is promiscuous, but whether or not that DNA takes and can be perpetuated, as Michael pointed out, is there are a whole nother series of barriers for that to work.
We've actually talked on TWiM about cases where bacterial DNA has been supposedly taken up into the human genome.
Right.
That was one of the first TWiMs with the Neisseria.
That's right. And one of our listeners sent a more recent paper, which we may get to in a couple of weeks. So I guess we shouldn't be surprised by anything. All right. Thank you, Elio.
Sure.
Michael, you have the next paper.
Yeah, this one was a little less of a heavy lift than I was glad when I volunteered for the fungal paper, because it wasn't as significant of a lift as the one that Elio has just done. And I have done a lot of work looking at fungal contamination and of the built environment. And this paper is entitled "The Diversity and Distribution of Fungi on Residential Surfaces." And the bottom line is they principally describe, in a university housing situation out in California, the diversity and distribution of fungi on surfaces. And they principally looked at the sinks, the windowsills, and then they looked at the people in the room, and they specifically surveyed the fungi that were on their foreheads. And some background for future figuring that was included in the paper that I found absolutely fascinating buildings only occupy 0.7% of the landmass of the Earth. And now Vincent won't ever believe
too
this.
Too much.
Here in New York, that's not true.
That's not true in New York. This next fact that the authors included is that humans spend 87% of their time indoors. And my mother would never allow us to spend 87% of our time indoors.
Actually,
and I think,
she specified Americans.
yes, she specified Americans. Yeah, that is true.
Americans.
That is true.
So
And
we're the worst, right?
yeah, I'm not sure that's something to be proud of.
And 69% of this time that we spend indoors is actually in our homes, except for newly minted assistant professors who are spending that time in their labs. So the reason they did this study is because when we think of the risk posed by fungi, is that it's principally for the materials that the fungi are shedding. The fungi are much more fragile than bacteria, and they will break off their canidia and pieces parts, and they become resident in the air, and they move about. So they're allergens, and some of them can become toxins. And so many folks have sensitivities to these materials, and they develop allergies, or it's thought that sick building syndrome is a consequence of having products of the and I'm going to hate myself for doing this, but the fungiome, the fungi that are
Mamma mia.
Yeah, well, this is why ohm is so important. There's too many ohms, and having worked on looking at the fungi that are associated with the built environment, it's not as easy as bacteria to identify, because we, over the last 130 years, have developed very good culture-based techniques to discriminate E. coli from Shigella and all of the others, as well as staph and strep that are in the built environment. And so we can tell them apart. And so it's really hard to tell fungi apart. And you have to look at them under the microscope. You have to scan so many fields in order to discriminate. And they go through the common number of species that you typically see in the built environment. And fundamentally, what their central hypothesis is that the structure of the indoor air is a subset of the fungi that are principally living on the outside are actually coming in to the inside. And so it's a subset. So the fungi then, their growth inside buildings and on surfaces and on us for that matter, contributes to the airborne contamination or assemblages principally as a consequence of aerosolization. And one of the things that any basic microbiologist will tell you is growth is principally limited by the availability of water. And, they need moisture. And in fact, they talk as one of the limitations or their studies, or one of the confounders, is that some of the housing units actually had humidifiers, because, as we know, the minute you get off of a plane in California, you need to apply chapstick to your whole body, because you begin to lose water, and this is why they can make prunes in California so easily, I think, is because it's a very dehydrating environment. So fungi are hard to identify. So what these authors principally did, and the authors were Rachel Adams, Marziel, Mileto, John Taylor, and Thomas Bruns from the Plant and Microbial Biology Laboratory of the University of California at Berkeley, is they used a traditional approach that we just talked about. They, had rapid sequencing using the usual suspects of PCR primers, and that would amplify fungi, and they did high throughput sequencing, got a lot of data, and they found some really neat things that if you sit back and you scratch your head, it's not all that surprising. They got more biomass where it was wetter. And so the sink drains had the majority of the biomass, but it had less diversity from the perspective of the number of different types of fungi that they found. And that was then followed by the windowsills, which had the middle level of biomass, but it had a larger number of different types of fungi. And then the ones with the lowest biomass, but the highest numbers of fungi were on the foreheads of the individuals, which I found to be absolutely fascinating, that as the biomass decreased, the biomass of the fungus, you actually got an increase in complexity of the organisms that were attaching to us. And, it was just an absolutely fascinating study about how they went about this. The other reason I suggested earlier on about talking about best practices for taking care of doing this high throughput sequencing and figuring out which method type, about halfway through their study, a new piece of software came out, and they ran their data through the new software and found a little bit of a different result. So this is almost our friends in the clinical microbiology laboratory are always coming out with a standardized protocol so that each clinical lab calls the same microbe or the same antibiogram the same way, and they have the same set of standards. And I think that as this technology matures, those of us looking at the surveying of the microbial world in these different environments from a clinical perspective are going to really want to know, can you compare this study to the other study, and whether or not the frequency and distribution of the microbes that are found in the community is really an indication of what is actually going on, and whether or not it indeed does represent a clear and present danger to the individual living in there.
Michael, it's interesting that so basically in this setting, which I presume are dorms, right?
That's right. Graduate student housing at Berkeley.
So they leave their windows open and the fungi are coming in, and that's basically what you find inside and on the people as well. But I wonder here in New York City, as you know, and in many cities, most of the buildings are sealed. You can't open the windows, and the air is circulated and filtered. I wondered if you would, I suppose you'd find a very different fungal biome, right?
Yeah, and in fact, in the colder climates like Alaska or Antarctica, the buildings are actually sealed so that you don't have air exchanges to the outside, because you lose too many BTUs with the leaky buildings. And in a place like Berkeley, I think many of the buildings are naturally ventilated. They don't have air conditioning like we do here in the Southeast, and where we do control for humidity. And that's an issue. And so again, I think they have contributed to the body of knowledge, because this whole issue of biomass and complexity, I think, is really one of their neat observations. They list the usual suspects you would expect for indoor fungal growth, things like Cladosporium, which is a big one.
You call it a weed.
It's the weed, and it is indeed a weed. When we were doing our studies, we did our studies at an Army barracks, the major principal training unit for the United States Army for recruits at Fort Jackson, South Carolina, and we did a 3-year study where we measured the indoor fungal population of new recruits, and we controlled for how often the windows were open and how often they were closed, and the humidity, and we measured the fungi from the outside. And it was a really hard study to do, because we did it the traditional way with culturing the fungi on Sabdex with chloramphenicol to kill off the bacteria, and we identified them. And so I still have more plates to go through and color plates in order to identify all the fungi that we found associated with the Army barracks. And so if I had, and this study was done about 3-4 years ago when we collected the data, and we're still reducing it from the, going through and saying, yep, this is Cladosporium, this is Malassezia, this is Rhodotorrhea, and it's so I really enjoyed this. We tried some of the modern methods as well. Biolog has a system out for fungi, which is based on metabolism, how the fungi eat stuff, and it's a redox dye. And it worked to some extent, but it didn't have sufficient discriminatory power that the human eye had in saying, yep, this is Cryptococcus, yes, this is Cladosporium, Cladosporiades. Its database wasn't sufficiently rich to the mycologists that we had identifying. When you have somebody who's got 30 years experience identifying fungi, they're much better than the databases out there. So I was really impressed looking at their fungal data that they were able to do and the visual representation of some of their multidimensional scaling based on their presence of taxa. So some of their supplemental data that they associated with the paper were really interesting for me to read. And I sent a bunch of notes to my technician that now is the time we have to reduce more of this data now that we have.
But to come back to Vincent's question about how it would look in New York City, I think the conclusion here was that the windowsills or the foreheads of people basically reflected a snapshot of what was in the air.
Right.
Largely because there's little replication.
Right.
So you just see the whole mess. It just sticks like flypaper. But even in New York City, then, in sites where you have moisture, you'd then get outgrowth, enrichment of a population, and then less diversity for that reason.
Right, and that's what they saw in the drains right here.
Right.
They also mentioned something that hit home. They say, the indoor growth of fungi indoors is generally not a problem unless you get water inside.
Right.
And that's what happened a year ago here in New Jersey. We had Hurricane Sandy, and we had this house down at the Jersey Shore, and we went down a week or two after the hurricane, and the water had receded, but one of our houses was full of, fungus growing on the rug and the walls and everywhere.
Yikes.
What a mess.
And you're never going to get rid of it short of putting up new sheetrock, which I believe you did.
We ripped out the rug. We ripped out the sheetrock. It's gone as far as we can get it out. Yeah, it doesn't smell moldy at all. And fingers crossed, maybe it'll be a few years before we get flooded again, right?
So Michael listed a number of species, and they all sound so beautiful coming off your tongue, Michael. But one that I thought was neat, they pointed out that they found ergot. Is that how you pronounce it?
Oh, yes.
Which is a plant pathogen. It's typically found on rye, but some of our audience may be familiar with it. It's actually the source of LSD. It can generate toxins.
How would you know that, Michele?
Because I had a delightful talk with Dr. Adams, the first author, who I'm going to tell you a little bit more after I finish this LSD story. So some of the secondary metabolites that this particular fungal pathogen or fungus that can produce can generate hallucinations, convulsions, and if you look through human history, like the first outbreak was described way back in 857, where it was called holy fire because of the weird behavior, but also people thought it was coming down from above. And then later, the Salem witch trials have actually implicated. In retrospect,
yes, implicated is the right word.
Perhaps it's one theory that because there's a correlation between how frequent witch trials occurred and climate change and the price of rye, etc., people can try to deduce whether the behavior that was viewed as witch-like was actually due to a poisoning, toxic poisoning by ergot or some other plant. But Elio, it sounds like you're dubious.
Well, I read the original claim, it was in Science actually. It wasn't a killer argument. I mean, it's a nice hypothesis, and
Poor
it's
people
plausible.
ate rye bread. That's effectively what they said. Is poor people ate rye bread that was contaminated with the toxin, and
there's a whole literature about that, and quite a few papers have been written gainsaying that hypothesis. So I have no stake in it. I wouldn't know, but let's be cautious about it.
Sure. So Rachel Adams, the first author on this study, she got her PhD at Stanford and was trained as an ecologist. In particular, she did field work in Utah, in Ogden, Utah, looking at the impact of diversity of plants and trees on the local ecology. And then for her postdoc, which is what she's doing now at Berkeley, she applied many of those same principles, but now is using communities that you can't see with the naked eye. She's become a microbiologist and is now mastering these computational methods that are so important in our field now. So she is loving microbiology and especially thinks this is an exciting time to be studying the microbiology of the built environment, houses. We've seen an increase in asthma. Can we relate that to particular hydrocarbons or volatile compounds made by particular fungi or not? She also, interestingly, is married to an environmental engineer. So that's an interest they share. And they're raising 2 kids. They've got a 4-year-old and a 2-year-old. And Rachel told a great story that she talks to her kids about her work and studying molds. And her one son was at a group playing with Play-Doh, and the adult noticed that there was some black fungi on the Play-Doh, and said, we need to throw this out. And her son protested and said, no, it's an experiment. We should study that mold.
Great.
So she's having a great influence on her kids even at that young age.
It sounds like her kids are having an influence on her.
They are. And actually something else we appreciated about computational biology, Michael, you made the joke in the beginning about the amount of time an assistant professor has to spend in the lab.
That's right.
Now that, with computational biology, you have a lot more flexibility where you do your work.
Yeah.
You need a computer.
You're writing your grants.
Sure. But whether you do that in your home office or at the lab, you're less tied to cultures and tubes and incubation periods.
The good old days.
Sounds like you had a good talk with her, right?
Yeah, she was great.
One of the things I noticed here, Michael, maybe you could comment on. They make this point that so the fungi in your home are mainly what blows in, or what grows in the drains. But in terms of bacteria in the built environment, it's mainly what falls off of people.
Yeah, and that goes back to the one I always get letters on, is that the average human is dumping 2 million skin cells an hour that are principally covered with Staph epi and streptococci and the other normal skin flora. And you're principally dumping gram-positives into your built environment. And I did a long study looking at the microbes of hospitals, and we don't think your house is very much different than a hospital, and it's principally Staph epi. 80% of the microbes are Staph epi. Jonathan Eisen is doing this very same thing along with Jack Gilbert of Argonne National Laboratory, where they're looking at the microbes associated with hospitals, and they're doing brute force big data sequencing to figure out who's there. So I'm going to look with interest and find out whether or not the biomass piece, as you look at biomass as a function of diversity, that may be an indication, because that really has a substantial influence in terms of pathogenesis. And, thinking about the whole issue of fungi being bad actors, I think they get a bad rap about sick buildings. I think we're just not doing enough fresh air into buildings, and the debris is just accumulating.
Michael, let me ask you a question. Cryptococcus shows up in abundance. That wasn't that. Are you surprised? I was.
Not in California. I think crypto. I wasn't all that surprised in terms of that.
It's a big time pathogen.
It's a big time pathogen, especially as you go further up the coast of our west coast. There's a new variant of Cryptococcus out there that's affecting healthy people, not just the immune compromised. And maybe we'll pick a paper on that new emergence of that. What's the species? Cryptococcus begins, I think, with an I.
Yes, that's right.
And and so I wasn't that surprised.
Is it Gad-eye that you're thinking about?
Yeah, Gad-eye. That's why I think it's an I. Yeah. So my brain and the neurons there's. But I wasn't surprised with Malassezia. That causes a rash, and the penicillium I wasn't surprised.
Dandruff, don't forget.
Yeah, the dandruff, the Malassezia is the dandruff.
One of the biggest infections of mankind.
Yeah. And the way we control it is with selenium, Selsun Blue. You add the selenium and it selects out and it selectively kills off the fungi.
Yeah.
So
I wish it worked a little bit better on mine.
Yeah, I did my PhD work on selenium, so I was called Captain Selenium there for a while. So I know all sorts of bizarre trivia on selenium.
This is a neat paper, which I think a lot of people can handle. It's open access. It's +1, so you can check it out and see the list of fungi that are probably in your home. And Elio, that's 2 weeks in a row now we talk about a fungus paper.
Yes, sir. Why not?
How about that, right?
Why not?
Well, that last one from last week, I'm still amazed that fungi are actually using radiation to make a living.
It's great.
It's incredible.
Yeah.
Michele, you have to check out that paper. It's a paper of, I'm forgetting our friend's name up at Einstein here.
Arthur Casadeval.
Oh, great. Yeah.
Arturo's paper.
I'll look on your website.
Check it out.
Website.
It's a very cool paper. And the materials were purchased at Trader Joe's.
It's in the blog if you want to look at it.
I actually showed that blog when I was out at WSU to a person who works in radiation. And I said, did you know that fungi can actually consume radiation as energy? No, they were shocked. And he says, I heard something about that. And I pulled up Elio's blog and showed it to him. So we had a nice conversation.
Right.
There is Elio brought to our attention not too long ago a paper published in June of this year, a letter in Nature, Topographic Diversity of Fungal and Bacterial Communities in Human Skin. So they looked for both, but they have a wide range of similar fungi in this paper.
It's a neat paper.
Malassezia, Aspergillus, Cryptococcus, Rhodoturia. So probably a lot of these came from the environment, right?
Yeah, I'm sure.
As we saw in this paper. All right. Very cool. I have a couple of letters that I think will be interesting to share. The first one is from a Dr. Robert Kelly, who writes, Dear TWiM Docs, I just finished listening to the latest episode of TWiM on the microbiote effects on obesity. So there you go, microbiote. There was discussion about the use of donor feces in the treatment of human disease. I thought this may be of interest. And he sends a link to a study that is currently recruiting participants called Harnessing the Healthy Gut Microbiota to Cure Patients with Recurrent C. difficile Infection. So if anyone wants to participate in that trial, look it up. Love the show. Yours truly. The next one is right up your alley, Michael. This is from Nick. I teach high school AP biology and microbiology and have a question about ATP synthesis via oxidative phosphorylation in gram-positive bacteria and I guess Archaea too. In mitochondria, chloroplasts, and gram-negative bacteria, it is possible to concentrate cations, hydrogen or sodium, in an enclosed space to drive ATP synthesis through chemiosmosis. I do not, however, understand how this is accomplished in gram-positive bacteria and have been unable to find a satisfying answer on my own. Some things I found seem to indicate that the cell wall may trap hydrogen ions. I read something about secreted enzymes not gaining function until they made it all the way out of the capsule due to the lower than optimal pH levels, or that aerobic respiration only occurs at high efficiency in the deepest layers of biofilms, something about protons concentrating when the attaching surface has a negative charge, which leads me to believe that gram-positive bacteria are indeed pumping protons out into the surrounding extracellular fluid in the hope that they can be used before they diffuse away. Microbiology is not my native discipline, but I have become enthralled in no small part due to the TWI family of podcasts as I try to both improve my microcourse and interject a healthy respect for our evolutionary forebears into the AP Bio curriculum. Any additional information you can provide would be greatly appreciated.
My goodness.
That's a deep, almost Talmudic question.
Is that right?
It's a really interesting perspective that he brings up. All living things pump ions for a living. From our cells, I guess you can view our mitochondria as being captured gram negatives, but everything has to move electrons, and the membrane is effectively where that electrical potential is moved. And the ATPases are what we understand in terms of pumping the proton out in order to facilitate. Remember, the membrane potential is based on two factors: the change in charge and the change in the chemical that is effectively creating the gradient. So charge is delta psi, and in the case of the bacteria, it's delta pH, or in the case of cardiac cells, it's calcium and other things. So in terms of thinking about the biofilms and this other stuff, I'm going to have to go and specifically look at some of the old papers from the 60s and 70s to see how the gram-positives, to actually look at specifically how they do it. Because I was trained in the gram-negative world, and I really have to go look at how the gram-positives do it. But they have to be able to do it because
Sure.
It's how it's the universality of energy. It's all membranes and gradients.
Okay. So no one else knows? Michele or Elio, you don't know the answer?
I do
No,
not.
I'm sorry, I don't.
Wow, I'm impressed. Or maybe
They
not.
stumped us.
Well,
Nick,
Michael.
Nick earns 10 points.
Yeah.
He stumped the group.
Michael, maybe next time you can have an answer.
Yeah, I'm going to go look.
All right, we have two letters from Jim, our friend in Virginia. First, just to pound on an older discussion topic of hand sanitation, here's a link to a recent study that shows gloves did not help reduce MRSA and VRE infection. This is an article in SIDRAP, which is based on a published study which showed that wearing gowns and gloves in all patient rooms in ICUs did not lead to a significant drop in combined levels of these two bacterial infections. But it didn't do anything bad either, they say.
Yeah, thank God for that.
Can you imagine them not wearing gowns and gloves, though?
The issue is, the bacteria are moving on the air currents. It goes back to that we're shedding microbes off of our skin, and VRE and MRSA. If you happen to be a carrier for those two microbes, will be shed off of your skin, and so they will move on the air currents. Just from walking through a room, you generate a substantial stream. Yeah, and so I'm going to go take a look at that one because that's sufficiently intriguing.
This came out in October in JAMA, and it covered ICUs in 20 hospitals in 15 states. This your kind of study, Michael.
Oh, joy.
Jim also sends in a second link to this site called Living to 100. It's a very interesting site where they ask you 40 questions, and then they tell you how long you're going to live. And it's run by an MD, supposedly. And Jim says He expects only another 10 years at best, and now apparently I will make it to 97. Ha. I have to try that, but I didn't have time to answer 40 questions. All right. This one, Elio, has to do with microbiome. Greg writes, Dear all, I have three brief responses to TWiM 66. 1, I always enjoy the episodes with all four of you best of all. Every episode is good, but the ones with all four are especially a treat. Thank you.
Isn't that nice?
Yeah, I thought you guys would like to hear that.
Thank you.
Yeah,
Two,
nice to hear that.
With regard to the subject of what should be meant by microbiome, I think that the word should refer to the genes rather than the organisms in a given environmental context. I do not think that this is what the word currently does mean, but this is what it should mean. There are two reasons for this. First, as Elio noted, the term is currently redundant over microbiota, and there is nothing gained by having two words that mean the same thing. The second reason is more complicated. Basically, it seems to me that it is possible for the word microbiome, considered as a catalog of organisms, to become meaningless when pressed too far. That is to say, E. coli K12 and E. coli O157H7 are very different organisms. The former has about 4.3 times 10 to the third genes, while the latter has 5.4 times 10 to the 3. Nevertheless, they both classify as E. coli. This means that when one speaks of the microbiota as a sort of catalog of the different species that live in a given environmental context, one slides over a lot of the diversity that is found there. To speak of molecules instead of organisms avoids this problem. Therefore, it is simply more useful to make the term microbiome refer specifically to molecules instead of microorganisms.
It's an interesting point. Well,
Yep.
I think we could discuss this for quite a while. I'm not terribly partisan of one or the other, but let me just say that it's only partially true that molecules are a simpler thing to deal with. Because after all, how are you going to say two genomes are different?
Yeah.
By what? By an operational taxonomic unit equivalent, an operational molecular unit? In other words, it sounds like different molecules are different, but by how much? And that's, I think, the same problem that we have with defining species. It won't go. The problem won't go away. So that may not be the main reason. The other reason, I think, that is a linguistic reason, that we have a perfectly good term, microbiota, and we could use that instead of microbiome to denote the species, or whatever one means by species. No, that's quite true. Well, I think that the listener is very astute and has pointed out more problems.
Yep.
Well, I mean, just consider what a prion is. A prion is a protein that when it folds into an aberrant state, it becomes a pathogen, if you will, that is thought to have the ability to change the way it's interacting with its host. And, is that the oldest form of, and I hate to use this word, life? So, now we're getting into philosophy.
Have to be flexible, that's
all. We've been there all along.
Yeah. His third point is Michael has a good point about the need for a word that covers both the microbial and the host molecules that interact with each other, but this concept needs its own third term. Assimilating this third meaning into the already confused term microbiome would only further tangle up the mess that Elio is rather conscientiously trying to untangle.
Very true.
Yeah.
Every episode is a treasure. Keep up the good work.
Hey.
Nice guy.
Well, that's very high praise.
Nice.
And one more is from Andy, who writes, To my valued friends at TWiM. Having recently read a Watson book on DNA history, I was reminded of an old TWiM episode that included an old-timer who gave numerous stories about his work at Woods Hole and Cold Spring Harbor. The episode may have actually taken place at Cold Spring Harbor. Even though I could tell that the history being related during this episode qualified as precious, I was clueless. Now, having read this book, I would give my right arm to hear this gentleman tell his stories about these labs in the 50s, 60s, and beyond, but I can't find the darn episode.
It was the video. It was the video that you guys did at Cold Spring Harbor with Stanley.
That's right. It was number 40. Can you please help? I will send you my right arm.
Please don't.
Don't do
Please
that.
Don't. It's covered with microbes. We know that.
Yes. Your avid fan and listener, Andy. P.S. It's my own fault, of course, that I don't have all your episodes. Most of them continue to fill up my phone and laptop and always will. I realize I can access the few that I reluctantly deleted. Isn't it a law that I would be looking for the rare deletion? P.P.S. I will spare you the hearty approbations that your show deserve. You must get those all the time. TWiM gives meaning to my life. Literally, I guess. That's nice. Yeah, that was episode number 40, a mecca for microbiology, and he is no doubt referring to Wacław Schabalski, who we interviewed on that episode. And that was me and Stan Malloy. And our myxobacteria friend from Iowa. What's his name, Michael?
John Kirby.
John Kirby. You know him?
I do. Well, I've heard him speak.
Yeah, he was fun. All right, that will do it for TWiM 68. You can find this on iTunes and also at microbeworld.org/TWiM. And if you have questions or comments, we love to get them, especially if you praise us, of course, but you don't have to. You can ask tough questions.
Shameless.
You can criticize, and you can say we got it wrong, too. We love all of them. TWiM, TWIM, at twiv.tv. Michele Swanson is at the University of Michigan. Thank you for joining us today, Michele.
It's a pleasure.
Neat stories about ergot.
Yeah.
Ergot. It was on the forehead, by the way, of those people. We have ergot in
so
Berkeley.
Don't be licking foreheads.
I think in Berkeley, it makes sense that they have ergot on their foreheads, right?
Well, at least LSD.
Yes. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent.
And Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. My thanks to the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.